All right, can everybody hear me?
We can.
Okay, perfect. So this is, I know you're probably wondering how 3D printing has anything to do with open educational resources, but I'm gonna present to you about these cost-effective 3D printed models and how we use them as a learning tool in an introductory biology course. And I'll also, tell you how this is related to open educational resources in a moment, okay? So this is actually, where'd my mouse go? This is actually a Bambu Lab A1 printer. We have two of these. We have two of another type of Bambu printer. This is just an example of what you can print out. So this is actually a model of DNA that you can take apart and put together. And it's one of the models that we used in this little pilot study here. So Christos isn't here. I think he's teaching right now. So I'm just going to give the whole presentation. Just wanted to let everybody know. So from the start, I'm teaching biology. We teach biology courses. And if anyone has taken biology before, you know that there's a lot of conceptual concepts that are difficult to visualize. So the traditional way of teaching biology is, you put up a PowerPoint and you put up some words, you put up some pictures, and you're basically asking students to use their imagination to try to put these pieces together to understand those concepts. And so what we're trying to do here is we're trying to implement the use of actual hands-on models. But an issue with that is purchasing these models, they're very costly. Just buying one simple model could cost you a couple $100s. And if you have a class of 20, 30, 40 students, it's going to be difficult to have that one model for all these students. So what we've done is we've taken these 3D, here we go. So we've taken these open resources, these 3D models that other people have designed. And so these are two websites that you can just click through, and you'll see there's lots of different models on Thingiverse, spanning from arts all the way through to the sciences. And then we can actually use these free models to print them and use them in the classroom. If we want to get a little bit more detailed and in depth, we can go to the NIH. They actually also have a 3D model repository where things like proteins can be downloaded and also printed. So for this, we actually received an NSF grant for these pilot studies. So this is just one small part, one small component of that, this larger grant of our pilot studies. But these are three models that we actually started with. So this is an actual model for protein translation. So how do we go from basic DNA to arriving to the protein machinery that does all the work in a cell? So this is actually the most interactive model that we have. So over here, there are nucleotides. This is supposed to be the ribosome. These are the amino acids, and this is the tRNA. So if you have no idea what I'm talking about, this is a pretty complex process. And so we've placed them on a magnet board, and then students can actually maneuver these 3D printed pieces around. This is that model that I showed you at the very beginning. This is just the basic DNA backbone and nucleotide pieces. It's supposed to help students understand DNA replication, and I'll come back to why this model wasn't so good towards the end of this quick lightning talk. And this is just a model of what the cell membrane looks like. This one is the plainest, most basic model, the least interactive. Okay. So so the design of this pilot study looks something like this. Okay, we give the students their basic traditional PowerPoint instruction. We do add a little bit of Q&A for some active learning throughout the lecture, because if you just keep talking, students kind of zone out. There's also an assigned textbook reading. And then at the end of all this, we give the 3D printed model, and we have the students play with the model and see if they can take what we just taught them and apply it to the model that they're looking at. And after all of this, we give them a non-graded assessment. And the way we sorted the comparisons is, so these are the three models we used. We had three core sections. So one section did not receive any models, so they only had the traditional PowerPoint and textbook reading, and then the non-graded assessment. The section 2 and 3, we alternated a use of a model to try to take the teacher variable, the instructor variable, and the student variable out of it. So section 2 would receive the cell membrane model, and then the next model, they would not receive it. And then the last model, they would. Section 3 would receive no model, yes to a model, and then no to a model. And just keep in mind, this is a pilot, and we're actually continuing to run these studies to get some more information, okay? So these are examples of assessment questions that were posed to the students for each of these topics. For cell membrane, these are 3 types of assessment questions. So what type of plasma membrane protein allows substances to move from extracellular to intracellular space? Which of the following are found in structure of DNA? What are the cellular components needed for the process of translation? Okay, so these are some of the questions that were given, and the assessments were about 10 questions in length. And additionally, they were also asked to rank using a Likert scale how useful the PowerPoint slides were, how useful the textbook reading was, and the last one is how useful the models were in their learning. That's if they received the model. These are the number of students enrolled that were answering the assessment questions and that Likert scale questions. The number of students varied because of absences for those days. And as we move towards the end of the course, so this is the beginning of the course, mRNA translation happens more towards the later end of the course, we start to get some withdrawals as well. So we do see some fewer students answering these assessment questions. But just to get into the results for the learning assessment, the first, so these are the learning assessment portions. So how many questions a student got right with them or without the model. For cell membranes and DNA replication, so these are the least interactive or the model with the least components that actually describe the whole entire process. There wasn't much change in terms of the assessment of getting questions correct. But the most interactive model, mRNA translation, that pretty much told the story of the entire concept that they were learning in that unit, actually did show a significant increase in the number of correct questions. So that's good. This tells us 2 things. The first thing is the models do work, but the models must be highly interactive and span a good amount of that topic that's being taught. And then student perception of learning with using the 3D model, which is the green, orange PowerPoint lecture, or gray textbook for mRNA translation. We actually noticed this trend that the textbook, nobody really likes to read. And we've swapped textbooks over the course of the past 5-6 years. And every single time we probe students using these kinds of questions, most of them, at least half of them, say they don't like to read a textbook. So I kind of look at this with a little, grain of salt here. The 3D models, they do find it somewhat helpful, but they find that the PowerPoint is usually the most helpful. For cell membranes, we also see something a little bit similar, except the 3D model, more students didn't find it as useful. They found it somewhat useful. But one thing to note that none of the students found the 3D models to be not useful at all. So they did find it somewhat useful in their learning, somewhat useful to extremely useful. And again, the textbook, they found it the least useful for learning cell membranes. The same, so DNA replication, we kind of have an overlap between PowerPoint, 3D model, and textbook. And I think that's because this topic is very intense. There's a lot of pieces that students have to put together, and the 3D printed model that we used didn't really encompass a lot of those parts. So these are some things that we are working on in this next unit in continuing this study. So in conclusion, students generally enjoy these 3D printed models, and we're definitely hearing that now that we're giving them more interactive models. So more interactive models yield greater learning gains and more positive feedback. Students prefer instructor provided PowerPoint lectures. They don't actually like the course textbook, but like I said, we've changed to OER. We used very expensive textbooks. We used very cost-effective textbooks. And overall, it's always been a trend that students don't really like the textbook. Right now, for in progress and future directions, we are increasing our interactive models. So right now we have a new DNA replication model that encompasses all of the pieces of replication, also transcription. We've also created mitosis meiosis models, which we've just piloted and acquired some new data. So hopefully next year, I get to present more on this. We are also looking we've also started implementing these project-based 3D printing where students can design their own models and then upload it to these repositories. So they're designing, they're having a sense of ownership, but they're also creating new OER works to deposit into these repositories. So 15 minutes are up. So I just want to acknowledge the bio department at our SUNY Old Westbury, because I have been getting these instructors that have been teaching these courses to implement all of these new ideas and technology in 3D printing. Our lab technicians for going along with everything that, that I've been doing. Also, I forgot to put here my lab TAs, so that's another component of this. My student teaching assistants, they are being funded by this NSF HSI award. And they're actually maintaining my 3D printing room for other students to come in and do their prints, but they're also helping me to design a lot of these interactive models for use in the class. So there's a lot more coming in the future. So I'm just going to stop here. Thank you for listening.
Thank you, Kenning. We appreciate your presentation. If you could take it down, we'd appreciate it. Thank you. And questions, please place them in the chat. And we will, Kenning, if you could answer them in the chat, or we can email them to you, whichever you prefer. And if you wouldn't mind putting your email in the chat as well for follow-up questions. All right, so next we have Kobe Klein from Old Westbury. You want to go ahead and share your screen? We are seeing your presenter view. You might want to hit display settings and switch screens.
That would be a good idea. All right, how about now? What do you see?
We see the view inside of SharePoint. If that's good with you, that's good with us. It's up to you.
Okay. Well, in that case, thank you everybody for joining us this morning. My name is Kobe Klein. I am an adjunct at SUNY Old Westbury in the Biological Sciences Department. Hi, Kenning. And I'm going to be sharing with you today a little bit about my evolution course. So there we go. I took over this course post-COVID when we came back into the classrooms, and I had previously at a different campus been teaching an OER course and absolutely fell in love with the whole idea of it. And so I tried to make evolution as OER friendly as possible. I've never really gotten a definitive answer about whether the full course qualifies as OER, but I definitely try to include as much OER content as possible. I don't have a proprietary textbook that I use. Many of the classroom activities are taken from open sources. Now, when I was building this course from the beginning, I initially wanted to include a unit on scientific racism, but I had to cut it for time. But a couple of things changed within the last school year. One of them was that I restructured the course to free up a couple of days for instruction. And the other was that I got an email about a new initiative at the university. And this initiative is led by the Social Environmental Justice Institute at the school. And the Institute is a way for all the campus community members to, support the university's social justice mission. And the new program, which was called the SEJI or SEJI Sections Program, was supposed to provide a framework through which faculty could really integrate issues related to social and environmental justice into their coursework. And I thought this would be a perfect opportunity to to get my scientific racism unit back into the class because uh, of a couple of different reasons. One is that obviously it's good to have as complete a picture as possible about the history of the study of evolutionary biology, including areas that we might consider dead ends. Uh, Beyond that, I also thought it was important that we spend some time in our second and third units about the history of evolutionary thought and evidence for evolution, that we uh, spend some time refuting arguments against evolution. And we do that because even though Darwin won and evolution is accepted by nearly everyone, there still exist people who believe otherwise, and it's important to know what they think and what they're getting wrong. And it's just as important to spend time talking about scientific racism in our class because there still exist people who try to use scientific or pseudoscientific ideas to justify their discriminatory beliefs and practices. And as aspiring scientists, the students should have the tools to refute those beliefs, not just from a moral standpoint, but from a scientific one as well, because scientific racism is bad science, and we should all know exactly why. So I thought for the rest of our time today, I would try to run through the lesson plan with you. I started running this class as a flipped classroom a couple of semesters ago, where I pre-record lectures and post them to Brightspace, and the students are supposed to watch them before class and come prepared. And so I integrated our unit on scientific racism into the larger unit of the history of evolutionary thought, which is our second unit. There are two other parts to it. The first part covers the history from ancient times up until the scientific revolution. Part two covers Darwin to the modern synthesis, and then part three covers 5 different aspects of scientific racism. And in the first section, we really talk about the evolution of the concept of race from its early days in 18th century Europe. We talk about the historical debate between monogenism and polygenism, and we talk about how those ideas gained acceptance and changed in the U.S. and in Europe in the post-Darwin era. And then our second part of the lecture covers the history of eugenics and the theory behind it, the pseudoscientific theory behind it, and we talk about how it was practiced both in North America and in Nazi Germany especially. Our next section covers race and intelligence, and really deals with the history of IQ testing, and how IQ was used to uphold racist ideas and to discriminate against many different types of people. And we try to debunk some of the myths about race and intelligence, and that's really the main thrust of it. I also wanted to especially talk about social Darwinism, which is not science per se, more social science than natural science, but it is based on a misinterpretation of the phrase survival of the fittest. And that misinterpretation in the early 20th century was used to justify the mistreatment of the poor, of minorities, and to support genocide. And then we wrap things up in the lecture by bringing things into the modern period, because as I mentioned before, scientific racism really isn't dead. It is a much more marginal ideology now than it was historically, but it does still exist. It's kind of rebranded itself to race realism. But there definitely still exist people who believe that there are biological differences between the races, and that those account for the outcomes, the social outcomes between these different groups. And so we touch on that a little bit in lecture, but we save the counter arguments for the last section of our talk, which will be about our lab. So when we get into the classroom, we focus on student-centered activities. We start with a little ungraded quiz on Kahoot based on the lecture based on the lecture. And we move into discussion questions, and these are a few of the discussion questions that we have. They're really open-ended questions that touch on the place of racism in science, both historically and presently, the nature of stereotypes especially. And the last part of the classroom activities involve a case study. And now I'd like to really put in a plug for the National Center for Case Study Teaching in Science. It is a fantastic resource that I use extensively in most of my classes. And there is a case study for just about anything you can possibly imagine, including one on the nature of race and where those concepts about race tend to fail. So the case study deals with the origin of skin pigmentation, as well as touching on the connection, or really the lack thereof, between pigmentation and other traits. And the point of the case study is to demonstrate the superficiality of skin color as a defining trait, as well as to point out the independence of other traits from pigmentation. And then the last part of our talk today is going to be about our lab. And the lab was, it actually started out as a homework assignment. And I expanded it into a full lab this past semester. And I asked students to read 2 pairs of journal articles and answer questions about them. The first pair of articles are about race and intelligence. And the first article that I asked them to read actually gives the results of a genome-wide association study, which suggests that there might actually be differences in intelligence between different population groups, and the authors are not explicit that they mean different racial groups. But if you know anything about the authors of the paper, it's pretty clear that is what they mean. And then we read a different paper which refutes the first paper, refutes the ideas that there might be any connection between race and intelligence. And we try to get at the distinction between genes in the environment and the relative contributions of each, as well as touching on the trouble with over-interpreting genome-wide association studies, which look at entire genomes of of different individuals, human individuals in different groups, and tries to associate different outcomes with different groups of alleles. And those are very, very ripe for overinterpretation and misinterpretation. The second pair of articles that we read are about a topic that I had never really contemplated before doing the research for this lab, which is how prenatal genetic screening might be considered a form of eugenics. Because some people might feel compelled or might feel pressured by their physicians to terminate pregnancies if there might be some kind of genetic abnormality, and that potentially could lead to a what's the phrase I'm looking for? Could lead to very negative impacts on society, or people thinking, it could encourage people to think even more about disabilities as being something that's negative and should be eliminated. There are reasons to believe that, that's not really a form of eugenics, but it is definitely a conversation worth having, and so we do. So that really forms our lesson plan, and I would love to take any questions if there are any.
Hi, Kobe, just to let you know, we are running up against the 10 minutes for your presentation, and I have Thea going next. So
Okay.
if anybody has any questions, if you could put them in the chat, I would appreciate it. And if you could answer them there, and if Kobe, if you could put your email in the chat as well so people can contact you.
I will definitely do that, yes.
Okay, perfect. All right.
Thank you, everyone.
Nadia, Kobe, if you could stop sharing so Nadia can. I appreciate it.
Thank you. Okay, can everybody see it?
Yes, we can see it.
Thank you.
All right.
Hello, everyone. My name is Nadia Joseph. I am from the nursing department at Nassau Community College. And I'm here today to present to you how immersive virtual reality is very important in nursing education. Why? Because it certainly which close the gap between theory and practice. So during COVID, I can move my slide. All right, during COVID, as we all know, we all were affected tremendously. So again, a lot of people, which is faculty, nursing faculty, you name it, experienced nurses, they all sort of like decided to have a different view on healthcare. Some of them really leave the healthcare, early retirement. And of course, this is the time that we struggle with clinical placement, meaning that the hospital will not allow students to come to the hospital. And of course, we didn't have any preceptor for capstone students. And absolutely, we're all aware there's always a major national shortage of nursing faculty. So for that matter, thank God for technology. So again, IVR, which is the immersive virtual reality, as you can see, is sort of like really preparing the student. It could be newly graduate nurses. It can be working in a hospital. It can also work in academia. So both places is a win-win situation. So it's real life clinical scenario. So at least students, in case they didn't have a chance to really, because they're not really exposed to every scenarios in the hospital, or they're not exposed to every scenarios in the lab either. So for that matter, this will give them an opportunity for them to really simulate their critical thinking. And as you can see, they have the headset and the motion tracking, so they can do it. They can use it for anything, medication administration. They can use it how to do CPR to make sure that they know how to do it very, very well. Whatever situation, as long as it is programmed, so the student or the newly graduate nurse will be able to function properly. Again, what is the purpose of that device is to really reinforce the classroom learning. For example, they can go and apply it because as you know that we teach in the classroom, but the best thing is for the student to be able to apply the theory. And it gives them a tremendous experience. It stimulates their critical thinking. It reduces the anxiety because, you know, as adult learners, so that device also gives them feedback from time to time. So they don't have to rely on the faculty to make sure that they know exactly what they are doing. So it increases their confidence in making sure that they are competent to provide care for their patient. Absolutely, it is a safe device. It can, the student or the learner or the newly graduate nurse can repeat that constantly to make sure that they are competent. That device provides feedback, so they don't have to wait for the faculty, so which is the best thing. It accommodates diverse learning style because some of us can see, you know, I don't have to say you all are faculty, increased engagement and confidence and exposed to rare or complex cases. Complex cases meaning it could be trauma, it could be blood transfusion, depending on their level. Because taking a student to a hospital is usually 8 students to per faculty. So that tells you that absolutely the faculty will not have the opportunity for the student to view or to be exposed to every complex scenarios. And again, it depends on the level of the student. It improved their clinical reasoning. It sort of like improved higher retention rate because again, they don't feel like they are stressed. They don't feel they don't have that much of anxiety, so they can stay and achieve their goals. It absolutely improved their psychomotor skills, depending on what the program, what is programming in that device. For example, if they do the simple skill, which is donning and doffing, the PPE, then in that case, it's sort of like guided. If they administer a subcutaneous medication, then like a heparin or insulin, so at least the psychomotor, or maybe administer IV fluid. So always, we're gonna always start from simple to complex. So I'm trying to like talk fast, so because I know we're running out of time. Again, like I mentioned, emergency response training. So this is a real world application that you guys can do. It can do any kind of scenario. It does not matter, whatever. It could be in a different discipline. It could be in the behavioral health. It could be maternity, whatever it is, as long as the learner is suiting the learners, making sure that the learner is competent. And also we have to be mindful of diverse cultural competence also, because we have, as a healthcare members, we have to be able to talk to different folks and accept their cultures and taking care of them the proper way. Interprofessional collaboration, this is a challenge in the healthcare industry, especially in the nursing field. How students sometimes they are afraid to communicate, they don't have that communication skills that also can use that therapeutic communication, for example, for end of life, or for trauma, how do you communicate with a family member, or how do you communicate with anybody, even the coworkers. Well, the thing is, it is very expensive. So the goal is for us faculty nursing department to write on grant and to make sure that we have the funds for us to have that supplies on board. Because other than that, it's gonna be a challenge. We need faculty on board, we need the buy-in, and again, when we do have everything, then we can integrate it into our curriculum. And just so you know, everybody, is the IVR is a powerful tool which really bridges the theory in practice. The students love it, they really love it because it's technology, they really love that. They enhance their learning, and it's that they can repeat it as long as they want. It prepare them for clinical when they go to the hospital, so at least we know because they do a lot of hands-on. And honestly speaking, it is a step forward future. Even the hospital is using it. I work at Mount Sinai South National also, and we are using it, like the hospital in the Nassau County in Long Island. Some of them are using it or are trying to buy the equipment. And then again, this is my first time here presenting, and I'm very pleased. And I thank you again for giving me the platform to share with you how IVR is a very, very important tool for any department to have. Thank you so much for your time. And thank you for listening.
Thank you, Nadia. We appreciate it. That was awesome.
Thank you.
We do have one more presentation. Beth Heavey, if you would like to share your screen. And Nadia, if you could put your email in the chat so people could ask questions, we would appreciate it.
Chris, it really is okay. We can just break for lunch. I don't want to run into everybody's lunch. I did present yesterday. I was just on the schedule for today as well. So I thought I was supposed to do a shorter version today.
Oh, okay. All right. Then we're all set.
Thank you. Yep.
All right. Thank you, everyone. We will be going to lunch now, and we will resume at 1 p.m.
Thank you.